Welcome to this tutorial about ChIPnCHOP, an automatic pipeline for chip-sex analysis. This software was developed by Ana González Toro, José Vázquez Pacheco, and me, Joaquín Tamargo Azpilicueta, for the subject Bioinformatics and Genome Analysis at the University of Seville. In this short video, I will teach you how this pipeline works in a few words, and what inputs does it need and what outputs you may expect to have using it. So, in first place, what does it need? Well, it takes a genome assembly and gene annotation, and both can be found at databases. For instance, you may find this at ensemble plans. There's the FASTA samples and the GFF3 annotations, and you can directly download it from here with wget, for example. So you have downloaded, and then you need to have the chip samples, the chip-seq samples. In this context, you may have the chip samples and the control samples. In this example, we have named the chip samples chip 1 and the control sample, the mock sample, as input 1. So it is really easy. ChIPnCHOP takes the directory where this genome annotation and chip-seq samples are in a parameter file. Once you have specified these directories in this parameter file to ChIPnCHOP, ChIPnCHOP is going to index the genome by bow tie tubules. This is going to make the computational cost lower, and it would be easier for this software, for the algorithms, to work in the next steps. So once you have done this, chip-seq samples are going to be analyzed in quality controls like FastQC, for example, and those reads are going to be mapped to the genome index by bow tie 2 and are going to be converted to .bam files by SAM tools. These map reads are going to be stored with quality control files within a results file in the directory that you have specified in the parameter file. So once here, these map files are going to be merged together by max2 copy, either with broad peaks or narrow peaks, depending whether if you have specified that you are working with transcription factors, in which case you may be working with narrow peaks, or if you're working with some specific epigenetic marks of histones that are more broad in the way that they interact with DNA. So in that case, you will be talking about broad, but generally talking, max2 copy is going to be taking the narrow, unless you specify the broad option. So once you have done this, the pick file that you are going to obtain is a .bed. This .bed is really useful because it will take the Homer, a program that will analyze the motifs of the .bed files, and a gene ontology enrichment that will tell some biological processes that are taking place within the peaks that have been annotated. And all in all, what you will have is a results file in which you will have quality control files, the map reads in .bam, peaks, geo enrichment, and peaks motif finding. This is all for now, but you can check more info about this ChIPnCHOP in our GitHub, github.com/jpaspa/ChIPnCHOP, and you may find some useful features in our README. I hope you have more profoundly understood how this software works, and hope to see you using it soon.